Yeah, thank you very much for the kind introduction. I know the title is quite long, but I didn't want to short it. So I have the fortunate position now that like all of you have already introduced your own data that we are presenting. So thank you for that. I want to take a quick moment in the beginning to talk about like what are actually those 3D tissue maps that we're talking about, like a quick definition. Niels showed it in the very beginning, but I thought it's good to bring it back now in the afternoon. So we're talking for 3D tissue maps that we start out with the raw volumetric data, and now already there it gets tricky. So we've seen that in the various talks. Sometimes you have a 3D volume out of the box, for example, from Lightsheet, but most of the times you have serial sections or serial sections where you do different imaging modalities like intertwined. So already there gets super tricky to just come up with the raw volume. So this is something also we as a community could maybe think about how to share our knowledge and help each other to get to this first step we are presenting here. And then as soon as you have this raw volume, you maybe also want to go in and do segmentations because they help you to actually now define what you want to look at. So as we've heard now from Sanjay, it's actually the same data. We want to look, for example, for glomeruli. And glomeruli are functional tissue units, and we can define how they're looking like. They're like roundly shaped, and they're embedded in this volume. And so you can use segmentation algorithms to define where they're located, and then use those meshes that you create out of the segmentation to go back and see what happens within them, between them, or how, for example, to interact with other functional tissue units like nerves, as we've seen right before my talk. So as soon as we have those two components, we can actually combine them with derived data, which can be numerical, like tables, but it can also be topologies or those like networks that we've just seen from Sanjay. And we have seen that there's like a lack of tooling and a lack of visualization to bring all of this together in an interactive way so you can make sense of your data. And the first stab at that we've presented in the HubMap data portal, which you can see here. And when I zoom in, you can see that we bring all the individual parameters together. So you have the raw volumetric data, you have your segmented glomeruli as meshes, and then you have some derived data, which is for now very simple. It was just the size of those glomeruli, but we can already think that we can bring networks into it as well. So when looking at this combination, we were able to identify some key challenges that we want to tackle with our visualizations. First, it was inspecting the general volumetric data. You want to get a sense of what you're looking at, which channels you have available, and how they co-locate. Then you want to explore some segmentation parameters. We know those segmentation tools are not perfect. Sometimes they need some tweaking. Sometimes they need to go back and you need to see, for example, set thresholds or whatever those tools need. So you want to do that and then see how well it worked. And finally, of course, you also want to inspect the segmentation quality. Sometimes for the glomeruli, we've seen that they merged, for example, multiples together. So you need to split them apart again. So you actually can treat them as individuals. And this also holds true, for example, for cellular segmentations. As soon as you've done that, you can now actually go in and derive some data with those meshes and analyze them together. And in some cases, we heard from our collaborators that they also want to generate new derived data interactively. So it could be here, for example, they want to add some measurements and sustain those measurements and then see them in the visualization. So in this first step, we found like all this data is 3D, and we've seen it now throughout the whole day. We present it always in 2D, right? It's just a projection on like on this flat screen or on your PC, but we actually wanted to do it in real 3D. So we thought like, how can we now go beyond the limit of a 2D display? And the way we are tackling this is by introducing a hybrid system, which we call Vitesse Link, and it combines mixed reality capabilities. I see there's one of the headsets over there from the Indiana group. So we're using the MetaQuest 3, which has a pass-through mode. So you still see your environment. You can look at your desktop. And because it has two displays, you actually get stereoscopic view and you can see depth. And with that, we've also gotten the feedback from the biologists that it's way easier to now perceive the 3D dataset in real 3D and get a better sense of co-location of things because of the depth perception. And as you were able to see in this short video, we can use this tool to interact with the data. We can just shift our head around and change the viewpoint. And we can also, for example, add derived measures in the same tool. So how does it work? On the top row, you can kind of see a depiction of how it worked in the past on 2D displays. You're just on your computer, you have your data and your derived data, and you interact with the mouse. And now in our hybrid system, we only bring out the 3D into the 3D space using mixed reality, but you can use both interactions. So you can reach out with your hand, but you can also still use your mouse. And as soon as sometimes, people get vertigo or, they want to get out of the XR world again, you can always work further on your PC because everything is synchronized. And that's where we've seen that we can bring the benefit of both worlds to the users and only use the XR where it made sense. And so what we're using as a hardware is a MetaQuest 3. I think now it's roughly $500, so it's quite achievable. It's not crazy expensive. And the standard consumer hardware like a MacBook Pro, already 2 years old roughly. And so coming from the 2D world, as you've seen before, this is a data set that Clarence and Peter Sorger showed before. So it's size-safe with multiple channels. We can toggle them on and off, and then we can see this 3D rendering here, for example, of this blood vessel that Clarence also mentioned earlier. And how does it look like now in the mixed reality? You do the same settings, everything on your desktop, and as soon as those two are synced, you can pop it out and see the real 3D floating here. And we heard very often that in XR or in virtual reality, it's tricky to do those, very intricate movements or the UI elements to bring them over is hard. So the way we approach it is you still use the UI on your PC that you're used to, and it is reflected automatically in XR because the two are synchronized. And what you can see here, that this blood vessel that Clarence highlighted before is actually a continuation of a larger blood vessel going through the collagen fibers. In an XR, you can actually see how it weaves through the 3D space, which is super tricky to even see here on the screen. So I invite you also to have the goggles with me. So if you have time later on, you can come by, you can try it out yourself as well. And so as Clarence mentioned also is they're doing segmentations as well. For example, here this blood vessel was of interest. Also, Peter Sorger said it. So you can bring this out and have a smaller field of view, but there you can see more details. And the way we do it is we actually embed the segmentation into the raw volumetric data. And now you can look at them together. So you can see how well the segmentation fits. You can also see the other channel information, which has not been segmented yet, and how it collocates with the segmentations. And you can do this all in XR. And the benefit is also, like I said, you can just reach out with your hand, you can grab it, you can pull it closer to you, and you can just very nicely inspect it and then combine it with some derived data for further analysis. So within our, sorry, that was too quick. Within our preprint, we're showing 3 different case studies of data from our collaborators, from Clarence, and also from Sanchez Group and from the Vanderbilt University. We've seen that before. It's the kidney data set that also Lucas from the University of Delft helped to pull together with MXIF. But in the meantime, we already got the chance to send out a handful, I think it's 7 goggles to collaborating labs so far. And then there are some people that have their own goggles and they also collaborate with us. So we're very fortunate to now have a large set of example data and people giving us feedback and, to have this continuation of people using our tool and then just giving us feedback where we can get better, where are the friction points, what are they using it for, and what do they appreciate in XR over a standard 2D display. And so you can see a lot of names that actually presented today. So I was always happy to spot some of the data sets that I've seen before being shown in our XR tool. Yeah, this is a preprint. You can find it on our website, and we are actively working on sending it out for review again. Now a little about future work outlook. I've shown so far how we can create these 3D tissue maps, a little bit about derived data, but now I want to show you a little more what we can do with them. As a first step, what we've seen is sometimes it can be cumbersome to set up your whole tool, define all the channels you want to see, and put this all into the test. So a very quick step was for us to think about ChatGPT integration just to ease the use of our tools. So what we did here is it was a project together with Arti. She was a summer intern from Hubble with us and continued her stay. So you can actually speak to our tool and say, show me my tumor and T cells. What ChatGPT does is out of the list of channels we gave it from the data set we're looking at, it chooses the channels that are representative of T cells and tumor cells. It tells you why it believes that. You can also change it. You can correct it if needed. and then it very quickly pulls them up in our data set, and it's very easy and quick to then view this combination of channels. And that's of course very beneficial if you're in XR, right? You can just talk to it and say, now I actually want to look at this or at something else. And it really works quite well, and it configures VUES automatically with these channels that you want to look at. Something a little more experimental was, we were thinking of giving ChatGPT access to statistics of the data. And what this made possible is I can go in and define a region here in my data and then ask ChatGPT or other LLMs what is going on in that area. And based on the statistics of the channels we're looking at, it can then start to kind of reason and tell us what it thinks is going on. So it tells us here, for example, there are melanocytic cells. So there's a melanoma present, which we already heard from Peter Sorger. And then it can also tell me that there are maybe some immune reactions in the same area. And the benefit now is that I can also ask about channels that I currently don't see because it has access to the statistics. So it can go through all the 60 channels I have available and tell me maybe there are other channels you want to look at because they're also interested in this and that way. Like I said, very experimental. So it would be now advised only for experts to being used. So it's more for like hypothesis generation, or just for you to get another view of your data that you maybe haven't thought about before. And what also both Clarence and Peter highlighted is often it's I think Peter called it the eyeballatron. So I think it's super funny that you can now actually go in and say, this area is interesting. Can you show me similar areas in my data? Or how often does this occur in my data? And then you can go through them and inspect them individually without having to like zoom and pan the whole data set all the time. So that's one. And then the other thing that was very much inspired by Sanjay, because we talked a lot about like 3D network analysis, and we've seen it now in their tool. And in parallel, we also tackled the problem together with them, and we took a more like visual approach. So we always try to have it in an interactive way. So what we're proposing is also 3D network analysis, same data as Sanjay showed. We're using their segmentation masks. And then we derived the network from it. And like I said, our approach was more to get it more interactive. So to help now, like, because we're working closely together, we can also see how well this can help to find maybe more motifs or find those in like larger networks. And as you see, we combine a 3D mesh visualization here on the left with some abstracted views of a node link diagram where you have both the nerves and the glomeruli in. And what you can see, for example, is when I highlight a nerve or a glom, you can see which other glomeruli I can reach via directly, right? Like this nerve, which glomeruli does it connect to directly, and which glomeruli I can only reach by hopping over another one. And that's also shown in this ego network that you see on the bottom right. So you can see who are my neighbors, like by which nerves am I connected to those neighbors, and which neighbors I can only reach by talking to my direct neighbor. And so this can then really help to, for example, find all those motifs that Sanjay presented. So I picked this from their paper, and we can see some of the motifs are quite easily spottable in our tool, like the lone glom, having no other glomeruli being connected to. Then, for example, a little more complicated, the hourglass. And now in this abstracted view, you can very nicely see there's kind of like a group of glomeruli. Then I have this central hub, which is now in green, that then connects to the other group. And all of this is like interactive. So you can zoom and pan, go in and really have a deep look into your data. And then also the grape that we've seen. So far for the kidney, another data set that Ashley Keeman shared with us at some time ago. They had the hypothesis, for example, she will talk about it right after me, so I'm not taking away too much about the biology, but there was some hypothesis that parts of the data, these pannins, which are proliferations in the ductal tissue in the pancreas, have more nerval innervation than normal ductal tissue. And so when we bring that into our network abstraction, you can actually spot that some of those pannins, the red dots, have more yellow dots, which are the nerves. But then we can also see that there's maybe some like still normal ductal tissue, which also has a lot of nerval innervation. So you could go back to the 3D rendering and see where this is at, and maybe this is already considered a pannin at some point. So we're very happy that you're here today and we like want to communicate and show you our results here. Like I said, this is work in progress. It's a work together with a master student, Benjamin Binder in TU Vienna, and I think he defends tomorrow. So we're happy that we could pull that together for today. Just very briefly about scalability. So Clarence shared with us their segmented cellular segmentations. And now it got super tricky because he just like showed us there are 11,000 cells, or in the bigger data set, 40,000 cells that you want to render and view at. And so we ran into scalability issues quite quickly. So the way we circumvented that is to use NeuroGlancer. It's actually the tool Connectomics uses a lot. So Jakob showed that before. And with that tool, we now have the right data formats to store all those meshes in like various resolution levels. So when you zoom in, you get the very detailed overview of those cells. And when you zoom out, you just get a glimpse of all the cells and how they collocate. And now, of course, this invites us to also go towards these analysis processes that we've heard before from Clarence. How do cells collocate? What is happening between cells? Who is touching each other? And what communities or bigger networks they form? And at the step that I'm currently showing, it's like randomly colored, but of course we're thinking of using like cell type annotations to really see a nice overview where they collocate and to go to these abstractions that you mentioned, like Peter Sorger is drawing or like, you know, pulling together in PowerPoint. We can now actually think of making this automatically at a large scale and analyzing these data sets much more efficiently. And then, of course, for the volume rendering, we're also looking towards dynamic multi-resolution 3D rendering. We know Imaris can do it, but we know there's a huge barrier in like using it often because it's expensive. And so we're trying to bring that also over into our like web-based and open source tools. So like our larger science community can actually use them. So to summarize, I've shown you some XR, AI, and visual analytics approaches that we can now like use together in these 3D tissue maps. Yeah, and I'm very happy for questions now.
Right. Are there any questions for Eric? This one over there.